This lecture is a broad overview of SARS CoV2 and the COVID-19 disease. I'm going to cover replication and genetics, a little bit of the virus origin controversy, testing and transmission, and a bit on treatment and vaccinations. So what is COVID-19? At this point, we're all pretty familiar with it. This virus has been around for about 2 years. The 2019 novel coronavirus infection is a respiratory infection caused by SARS CoV2. The word corona comes from the Greek word for crown, and so you can see in this particular model how the microbe has very prominent spikes that give it a crown-like appearance. These viruses in this electron micrograph illustrate that point even better, showing you the corona-like array of spikes around the surface. Now these spikes are used for binding to receptors, as I'll show you. The virus was originally identified in December of 2019 in Wuhan, China, and then obviously spread globally from there. This is just a little bit about coronavirus taxonomy, just to show you the range of common coronaviruses. In fact, the genus alpha coronaviruses consists of a variety of viruses, 2 of which are endemic cold-causing coronaviruses, 229E and NL63. The beta coronavirus genus contains SARS-CoV-2 and then its close relatives SARS-CoV-1 and MERS, the Middle East Respiratory Syndrome coronavirus. And then there's 2, in this particular cluster, 2 cold-causing coronaviruses, HKU1 and OC43. So as a group, coronaviruses are not novel. They, again, are endemic, and several of these members spread yearly, causing seasonal colds that can be fatal in a lot of individuals, particularly in nursing homes. These viruses use a aminopeptidase N receptor, and the betas use ACE2 receptor, which is part of the renin-angiotensin system that regulates blood pressure. This slide is a nice overview of the entire replication cycle, and we start here with number 1, in which there is attachment of SARS-CoV-2 to the host cell using its spike protein. And one of the things that the virus requires, that makes it more efficient at entering host cells, is not just the binding to ACE2, but also the virus requires a host-derived protease, in this particular case a serine protease, but there's another protease called furin protease that can cleave and process the spike protein. When you get this cleavage event, that allows the virus to then penetrate, causing membrane fusion event, in which the virus's envelope fuses with the envelope of a respiratory epithelial cell, for example, allowing the virus to enter and then inject its RNA, its plus-stranded RNA, into the host cytoplasm. This plus-stranded RNA can then directly be translated into proteins, which are then, primarily they're polyproteins. They're so long polypeptides that have to get processed by a virus protease into individual pieces. And then these pieces become, again, parts of the virus. They become structural proteins and so forth. The genome, the plus-stranded RNA genome, has to then serve as a template for full-length minus strands, which in turn can then be transcribed into full-length plus strands, or more genomes, in other words. One of the cool things about the virus, too, is that the virus, while it's replicating its RNA, will make subgenomic RNAs. And so these smaller RNAs, rather than making a full-length piece every time, the virus will make so-called subgenomic RNAs that encode very specific proteins. And so these pieces, these mRNAs, are translated into the structural proteins, the membrane and envelope and spike proteins and so forth, that embed into the endoplasmic reticulum, are then trafficked to the Golgi, where they're glycosylated and processed, and then eventually bought off of the Golgi into these vesicles, where they're joined by a full-length copy of the plus-stranded RNA genome, and then the fully formed virus, which forms inside the cell. It's pretty unusual. Many viruses actually form at the surface of a host cell, but SARS-CoV-2 forms in these endomembrane vesicles, gets released into the world, where it's then hopefully greeted by a robust immune response that will, again, trigger an enhanced immune response, and in some patients, an enhanced pro-inflammatory response, which can cause a lot of damage, the so-called cytokine storm. So here's a close-up of the entry and spread of the virus with specific emphasis on the role of its spike proteins. So the virus can actually bind to abundant sialic acid moieties on the surface of an epithelial cell. And so by binding to these, it gives it attachment to the surface. The virus can concentrate on the surface by binding to these more abundant sialic acids. And then ultimately that gives way to tighter and more specific binding of the COVID spike protein to protein receptors, the ACE2 receptor in particular, on the host cell. Once we have COVID spike and protein receptor binding, then the surface protease of the host cell, so the protease that our cells make, can process that spike protein and facilitate the membrane fusion that allows the virus's RNA to enter the cell. And then that's, again, replicated and so forth and translated and makes fully formed virus particles, as you see here, in those vesicles that I showed you in the previous slide, that can then release out into the world. This virus can then be sneezed or coughed or dropleted out into the world in some way, just through speaking, and infect others, or benignly land on surfaces and be destroyed. The other potential outcome here is the formation of syncytia. Syncytia formation is basically just membrane fusion between neighboring cells. And so in this particular case, some of these virus spike proteins find their way to the epithelial cell surface, where they can attach to receptors on neighboring cells. And when this attachment takes place between the COVID spike and ACE2 receptor, for example, of a neighboring cell, the surface protease can cut that and basically stimulate this response. But instead of the virus, whole virus entering the cell, in this particular case, it causes the cells to form syncytia, which is basically just a bridge between their cytoplasms. And that allows the virus to then pass directly into a host cell without having to go out into the world in the canonical way, go out into the world and reinfect. The virus's RNA can actually just move seamlessly to the neighboring cell. This is, essentially like a laterally spreading wildfire. The virus doesn't have to go out into the bloodstream or into the mucosa and potentially stimulate an immune response. The virus can stealthily just move from cell to cell. And that's predicated on, again, the interaction between these virus spike proteins and the ability of our proteases to cleave these efficiently. And this becomes an important concept when we look at the virus evolution issues. So here's another look at this, but with a little bit more detail in the immune response. So here's the virus, and you get to see a few more of the parts of the virus, the envelope protein, the spike protein, and membrane protein, and the RNA that the virus has, the plus-stranded RNA, and then this nucleocapsid protein, which protects the RNA. The virus binds to ACE2 and, again, uses the transmembrane serine or furin protease to allow it to enter the cell by membrane fusion. The RNA can then be released, and the RNA is, of course, translated into proteins, but also transcribed into more RNA to make both genomes and virus pieces. And then these fully formed viruses can leave and go out into the world, where they're hopefully greeted by a robust immune response. In this particular case, you see an antigen-presenting cell called the dendritic cell processing this virus, basically just destroying it and then displaying the antigens, the viral peptides, on its surface. That it displays those to a T helper cell, and the T helper cell then activates cytotoxic T cells, which kill intracellularly infected cells. And the T cells will also activate B cells, which can go on to make very specific antibodies against the current invader. When the immune response is working properly, And as I said, these cells, the cytotoxic T cells, will kill intracellularly infected cells. You'll get an appropriate antibody response to neutralize virus, and essentially the infection clears, either without symptoms or with mild symptoms after a few days, as is the case with most individuals. However, some individuals, particularly individuals with multiple comorbidities, experience a cytokine storm. The respiratory distress and acute respiratory distress syndrome that occur as a result of an overactive immune response that's ineffective, essentially. So here's the virus, and it's not just SARS CoV2, it's influenza and RSV and hantavirus. Lots of viruses cause a cytokine storm. The virus here infects an airway epithelial cell, and this infected airway epithelial cell will produce alarm chemicals referred to as cytokines or chemokines that attract immune system cells to the fight. So these immune system cells are moving through capillaries and so forth, and then when they detect a concentration gradient of chemoattractant, they push their way through these endothelial cells in order to join the fight. And each time one of these pushes through, it causes a little bit of leakage, which in and of itself is a problem because the capillary leaks and that affects blood pressure. But the immune system cells come to the fight, and hopefully they clear the infection, but in some individuals, they can't. They're ineffective, and it leads to them producing more cytokines and chemokines, which attract even more immune system cells to the fight, and these produce even more cytokines and chemokines, which in turn causes local inflammation, which is again the swelling, the fluid accumulation and production of clotting factors, for example, which go into the bloodstream and cause blood clots. So you get systemic inflammation that causes damage to the capillary epithelium and causes it to leak. You get blood clots, which obviously can withhold oxygen from various organs and cause tissue death. And of course, all the swelling and fluid accumulation that could even potentially drown an individual in their own fluids. One of the reasons for this cytokine storm is illustrated here. You have a low-risk individual that produces healthy natural killer cells, just as one arm of the immune response. This is the part of the innate immune response, not the antibody, but innate immune response. Healthy NK cells recognize intracellularly infected cells. They bind to them and kill them effectively. They produce an array of chemicals which can kill these and clear the infection. But a high-risk individual might have dysfunctional NK cells, and by that I mean cells that are not actively producing enough of the cytotoxins that it needs to kill these cells. And so you need a lot more of these to come to the fight and produce a lot more chemicals to affect any kind of result. But if they continue to produce too many chemicals, that's going to attract even more immune system cells to the fight, which in turn leads to an unproductive, overstimulated response, and all of those things that I had mentioned previously: blood clotting and inflammation and fluid accumulation and issues with capillary endothelium and so forth. We know that that dysfunctional NK cells are often a hallmark of certain comorbidities, such as advanced age and obesity, for example. So here's a little bit of a right turn, looking at the origin of SARS CoV2 and its close relatives, but it speaks to one of the issues that I mentioned previously, which is the efficiency with which the virus binds to human ACE2 and is cleaved by the furin protease, that an enzyme that our body produces. So looking at some of the relatives, it's clear that the original coronavirus probably came from a bat. In the case of MERS, the bat virus contaminated camels as an intermediate host, and then evolved, and through sharing of respiratory secretions went into the final host, which is a human. Humans regularly ride camels and eat them in this part of the world, and so there's pretty regular exposure between these respiratory secretions. This is a particularly deadly virus, so when it does jump into the population, it burns out fairly quickly. Here is SARS-CoV-1 trajectory, starting with the bat origin here and presumably passing through a civet cat. Civet cats are eaten, but they're also farmed for particular weird kind of civet cat coffee that that is made by passing certain coffee beans through civet cats, and then the processed beans that come out are supposedly a delicacy. The point is that humans in certain parts of the world have a lot of contact with civet cats, and so it's not surprising that there was a contact between populations and transfer. The big question, though, for SARS-CoV-2 is where did it come from? It's pretty clear that its closest siblings are bat coronaviruses, and there was an instance where a pangolin coronavirus had a high similarity to SARS-CoV-2 as well, but there were some very stark differences between these viruses, the pangolin and bat virus, and the human virus, which led individuals to conclude that there's some intermediate here, possibly another animal, but possibly something that was artificial in the lab. So if you watch this video, it's about a prominent professor in Australia whose research I'll talk about, but basically had suggested that it's very likely that the virus evolved in lab, possibly in humanized cell culture, possibly in humanized mice that are expressing human ACE2 receptors. And this allowed the virus to undergo accelerated evolution to become highly adapted to human infection and causing explosive infection out of nowhere. So here's a little bit of that research, and the researcher's name is Nikolai Petrovsky. Dr. Nikolai Petrovsky is a vaccinologist and a protein biochemist. And his paper, In Silico Comparison of SARS-CoV-2 Spike Protein ACE2 Binding Affinities Across Species and Implication for Virus Origin. Basically what he did was mapped the SARS-CoV-2 spike proteins against various ACE2 receptors in different animals, and asked the question, based on binding affinities and computer modeling, what does SARS-CoV-2 bind most tightly to? And he concludes, these data suggest that SARS-CoV-2 spike receptor binding domain evolved by selection on human-like ACE2. The spike protein exhibited the highest binding to human ACE2 of all species tested, which essentially defies evolutionary logic in this particular case. If the virus recently evolved from bats, it should most tightly bind to bat receptors. If it most recently evolved from pangolins, the virus should most effectively bind to pangolin receptors. Another odd thing about this, though, that speaking to the politics, was that this paper took about a year from original submission to acceptance to be published. And in this day and age, this type of important information should not have taken a year. In this day and age, papers are being pushed out so fast that they're not done. And so the fact that this one took a year suggests some really strong pushback against even the hint that this might have come from a lab. But there is genetic evidence for that, and I'll show you some of the lab leak evidence. Here's an overview of the SARS-CoV-2 genome, showing you open reading frame 1. These are just big blocks of RNA that are translated into polyproteins that have generally like functions. So open reading frame 1 encodes a lot of genes for suppressing the immune response and taking over the host cell. Open reading frame 2 has some genes that are necessary for replication and so forth. And then this back part of the genome is mainly structural proteins like the spike protein and so forth. But if we look at the spike protein, and more specifically this tiny little sliver, this tiny little region of the RNA, which I've highlighted here, you see something that speaks to an unnatural origin. There's the actual cleavage site in which the protease operates on, the human protease operates on, that must cleave the spike in order to give the virus super high affinity for entering human cells, falls right here in this cluster. And here are the codons: CCU, CGG, CGG, GCA, and CGU. And those encode the amino acids proline, arginine, arginine, alanine, arginine. And so this PRRAR sequence gives SARS-CoV-2 incredible infectivity for human cells. And when I showed this, nearly 2 years ago to a colleague of mine, he basically said, "Hey, that's odd. Human gene jockeys commonly use CGG for arginine. Is that a complete coincidence?" So basically what he's saying is that's really odd that there'd be two very rare CGG codons right next to each other in this super important furin cleavage site, suggesting that somebody recognizing the importance of a furin cleavage site might have artificially put this there using some of the signatures, CGG, CGG, that human gene jockeys often use. Just to show you how important this is, looking at the mutations in the Delta variant actually changed this proline, this CCU, to CGU, and that encodes arginine as well. So now the furin cleavage site for Delta variant is RRRAR. So the more arginines that are in here, the more polybasic, quote unquote, this region gets, and the more efficient the furin cleavage event is. And so that speaks to the success of the Delta variant by modifying this proline to an arginine and making it even more infectious. So what it looks like to me is that humans potentially put this here in order to create, quote unquote, pre-pandemic strains of virus that they were hoping to study their infectivity and design vaccines for before they even become pandemic strains. And the virus, in turn, leaked out of the lab and basically started evolutionarily modifying this region to make even more efficient versions of it to increase its own infectivity. One of the reasons why I think it was unnatural is because when you look at the closest relatives to SARS-CoV-2, they don't have anything like this, such as the SARS-1 virus, pangolin, RATG13, which is a bat virus, which was highly similar to SARS-CoV-2. In fact, probably the most similar virus to SARS-CoV-2, has nothing like the PRRAR. And in fact, when we zoom out and look at SARS-CoV-2, here's Wuhan HU1, that's SARS-CoV-2. When we zoom out and look at that against a whole bunch of similar viruses, highly similar viruses with very similar spike proteins, this is the only one with the furin cleavage site. Now, we know that there are coronaviruses that have a version of this furin cleavage site, but they're very distantly related. And it's, again, they're completely different viruses. And so some viruses are capable of recombination, but generally recombination only occurs in viruses that have a high degree of similarity. And again, viruses that that have furin cleavage are sporadically found in very distantly related coronaviruses. The ones that are most closely related, none have this furin cleavage site. So the question then is, what is the origin of SARS-CoV-2? Was this an accidental lab leak of a CoV-2 strain that was optimized by passage in humanized mice or cells? Is it an accidental leak of a deliberately genetically modified SARS-CoV-2 strain, which I favor this second bullet point? Is it natural transmission from an animal, a wet market, etc.? I don't favor that one at all, primarily because I don't think that there's evidence for it, but I also don't think that there's evidence for there being a lot of live animals at the market too that could have potentially sneezed in somebody's face and caused an explosive outbreak that just happened to occur a few miles from the Wuhan Institute of Virology, which we know was experimenting on coronaviruses and creating these optimized strains basically optimized for infecting humans. Who knows if there's another scenario? But one of the cool things is that we have to ask ourselves is, what are the ramifications of each of these scenarios? Certainly this would be a black eye to science and potentially even the Chinese government, and it's possible that that's why a lot of this is being covered up. What additional data do we need? Well, we need more sequences, as many as we can get. If there is a natural virus out there with a PRRAR furin cleavage site, then that could be all the information that we need to know, that these could occur naturally. We just haven't sampled that particular population. However, I would argue that we've sampled hundreds of thousands of coronaviruses in thousands of different kinds of animals and never found anything that's A, closely related to SARS CoV2, and B, has the furin cleavage site. So here I'm going to talk a little bit about the testing methods, molecular diagnostics methods using RT-qPCR. And these start with usually a nasopharyngeal or oropharyngeal swab, but we often will use saliva, which is easier than sticking a swab deep into your nose and potentially causing bleeding. Either way, though, you collect viruses, whether it's saliva or a swab, and perform an RNA extraction. You can actually avoid the RNA extraction in saliva by just heating the saliva sample at 95 degrees Celsius for about 5 minutes to loosen up the saliva and break open any viruses. But with wastewater, which is what I most frequently do, we have to use a full-on RNA extraction protocol that cleans up the RNA and separates it from all the filthy matrix. So then the next step is the RT-qPCR step. Once you have the purified RNA, in this particular case, you need a good clean RNA that's in good intact shape. You perform a reverse transcription step in which your primers and probes are mixed with an enzyme that will create complementary DNA copies of that RNA. And every time it does that, it adds a label that can emit fluorescence, and therefore the machine can detect the amount of retrotranscriptions that take place based on the amount of fluorescence that's formed. Over time, the number of reactions that have to take place in order for fluorescence to build up and cross over a threshold that you set on the machine. Basically, you're telling the machine anything below this threshold is negative and anything above it becomes positive. The question is, how many cycles, how many doublings are required in order to get fluorescence that crosses over the threshold? And this cycle number, this critical cycle number, or so-called CT, or cycle threshold number, is the essentially PCR score that you would get. And that's sort of illustrated here. Here's an individual with lots of viruses, intact viruses, an acute infection. Here's an individual, a non-contagious individual with very few viruses, mostly non-viable, just some random strands of RNA. And these are subjected to RNA extraction and then the RT-qPCR. And so this robust pool of RNAs is then doubled in various cycles, and with each cycle producing a measurable amount of fluorescence. The amount of cycles required to get to a fluorescence reading that surpasses the threshold is the CT value, illustrated here. Because this starts with a very low number, you need a lot more cycles in order for this sample to exceed the threshold. And so this might be a hypothetical data set for this circumstance. Person number one, this person here, might have a CT of 27, meaning this reaction required 27 cycles in order to exceed the threshold. Person two here, with few contagious viruses and low starting material, would require a lot more cycles in order to exceed the threshold, and therefore this person's CT value might be 37. This one would be significantly lower, and you can look at this and estimate who's sicker based on this number here. But again, that's not always the case. What's difficult about this, and in terms of interpretation, is our inability to know what this means in terms of viable virus, or what this means in terms of viable virus. And I'll leave the quote from Dr. Fauci to you in order to see what I'm talking about in terms of confusion. Now, when you get a report from a RT-qPCR, it typically just says detected or not detected. So it doesn't tell you whether you're this person or this person. And sometimes your symptoms might, but often not. And as a result, you could be quarantined and subject to all kinds of contact tracing rules and so forth without really knowing what your CT value is. If you get a cycle threshold of 35 or more, the chances of it being replication competent, in other words, a true positive, are minuscule. You almost never can culture virus, that is, detect a true positive result from a 37 threshold cycle, even 36. This is from Dr. Fauci himself in this particular interview. So results should really be presented as not as positive or negative, but as cycle thresholds, so we can get a properly defined case. And I'll just give you some practical examples of this, how it can cause problems. This is from a former student of mine who's now a medical doctor. And he basically says, those pesky strands of RNA up the nose create problems for us, as patients going to nursing homes can't be discharged until they've had 2 negative PCR tests. Our own guidelines won't let us retest a patient who has been in ICU until 21 days after a positive. So even if they've completely recovered clinically, we still get positives. And it's not unusual for one test to be negative and then the second 2 days later to be positive, which keeps them in the hospital and makes me wonder how that is influencing counting hospitalizations across the country. They're likely no longer infectious, but kept in the hospital in isolation for an extended period, and then they get lumped into the entire cohort, again, of hospital overcrowding or that kind of thing. And here's some quotes from various papers. This individual says, qPCR results can remain positive for up to 5 weeks after the onset of symptoms. So again, individuals just keep testing positive because their body keeps producing or sloughing bits of potentially non-viable virus. This is another quote. The RNA could be from non-viable or killed virus. Live virus is often isolable only during the first week of symptoms, but not after day 8, even with positive RT-qPCR results. So, one way to circumvent this is to come up with an approach, a live virus quantification assay. And so we often will do a version of that called a Plaque assay, in which a sample containing virus can be diluted, and then dilutions of the virus of the potential virus containing preparation can be mixed with cell culture. And then we would look for cell death on these cell cultures plates as an indicator of live virus. Here's an actual picture of that. And you can actually quantify these little spots, which represent where the virus was deposited and killed host cells. So the question is, how can we make this into a rapid assay to be able to correlate a specific CT value with a person's load of viable viruses? As far as transmission and really getting the correct perspective on this pandemic, we really need to know who are the super spreaders, who are most affected by the virus. And we really genuinely know that. But what surprises me is that we haven't been able to tailor our public health messaging accordingly. Instead, we create these blanket policies which keep everybody suppressed, and over time that causes a tremendous amount of erosion and collateral damage. We know that, for example, nearly 80% of COVID patients hospitalized in the US are overweight or obese, which should have allowed us to create more tailor-made public health policy. We know that a lot of individuals that died of COVID had at least 4 comorbidities, according to the CDC. And again, that should have allowed us to create more tailor-made policy, such as allowing young kids to go to school as opposed to letting them become another array of collateral damage, mental health issues, learning issues, and so forth. Speaking to what I just said previously here, this data set from a PNAS paper shows the correlation between super spreading and BMI years, which is body mass index times age on the x-axis here. So again, we're looking at particle emissions as a function of BMI years, and they're using data from databases to assess BMI. But obviously, as you can see, there's a strong correlation. And again, I'm not trying to single out or shame anybody in particular, but we know what the virus is. We know who the primary targets are, even though it can vary, and there's evidence that other individuals that quote unquote didn't have any obvious comorbidities have succumbed to the virus or at least been really sick. But we still have to understand who the vast majority of the individuals are, and again, tailor our policy accordingly. But what we did was kind of the opposite of that, where we know that BMI and those types of issues can adversely affect your immune response and in turn make you more susceptible to the virus. And what we did was created a policy which actually exacerbated the problem. Up to 10% of the population said they gained 50 pounds or more. The average was around 30 pounds of weight gain because of the lockdowns and forcing people to stay home, not hike, not go to the gym, not go to the beach, and so forth. And that obviously, according to what we know, was not good. It was actually the opposite of what we should have been doing in this particular pandemic. Another thing that, again, another piece of evidence just came out recently from Johns Hopkins showing that lockdowns really only reduced COVID-19 mortality by about 0.2%. Lockdowns should be rejected out of hand, and that's something that a lot of us are saying from the start and receiving a lot of pushback on. But I think that, now that the politics have changed a little bit and people are starting to get a little bit tired of this after 2 years, being constantly told to hide, and new variants are popping up, and there's the new variant, and we need to keep hiding. And we've gone from 2 weeks to flatten the curve to 2 years. I think people are starting to really wake up. And as a result, the messaging is changing. It's just a little bit about prevention and treatment. In this particular case, some of the emphasis is on over-the-counter stuff, the easy things that we can do, such as getting out into the sun and getting a little bit of vitamin D, or actively taking vitamin D3. There's a very well worked out mechanism for why this might be helpful to a COVID patient, because it might counteract the pro-inflammatory reactions that lead to a cytokine storm. In this particular case, the vitamin D3 is converted to 125 hydroxy vitamin D, which in turn interferes with the NF-kappa B pathway. So this transcription factor can activate some of these pro-inflammatory molecules, such as tumor necrosis factor alpha and interleukin 6. So again, this has there's a mechanism for this, and now what we need are more robust studies to support or refute the potential role for vitamin D3. But if something as simple as this could be the antidote for a lot of suffering, we should definitely explore it. Anti-inflammatory drugs such as glucocorticoids and dexamethasone, all these things also have potential mechanisms in blocking NF-kappa B and cytokine storm, and clearly should be experimented and tried. In many cases, they have been, and a lot of people stand by a lot of these, such as budesonide and dexamethasone. Antiviral drugs have been really controversial. The antiviral drug remdesivir, for example, is standard of care, but it's questionable whether it's effective. It's extremely expensive, which in my cynical mind is probably why it's been considered standard of care, despite very poor performance. One that's been very controversial, hydroxychloroquine and ivermectin, are very affordable, very cheap off-label drugs that have been used off-label to treat COVID-19. But again, these have been very controversial, and my cynical side wants to say that they've been so controversial because people making a lot of money off of something like remdesivir at $3,000 a treatment are not going to be super excited about selling, $1 a pill hydroxychloroquine or whatever it is, $1 a bottle, who knows. But again, the point is not that these work or don't work, it's that we need to give them a fair shot. And so, and obviously I'll get into a little bit of antibody therapy, monoclonal antibodies and so forth, that that would serve as a kind of passive immunity where you get an immune response from one animal, and then you harvest antibodies from that individual and then infuse those into a sick patient to bind up virus and get rid of it. And then obviously vaccines. Vaccines have been quite a game changer overall in the pandemic, giving a lot of people not only protection from severe disease, but also peace of mind. And I'll get into both of those things. There's a little bit about remdesivir. Remdesivir is actually a prodrug, and it's in this particular form, our liver enzymes and so forth will process this into a molecule that's an adenosine nucleotide analog. So it looks like an adenosine. Here's the ribose sugar mimic, and here's the adenosine. And here's the 5'. This is a 5' hydroxyl, but it gets phosphorylated to become a 5' phosphate. And here's the 3' hydroxyl, which is essential in replication. But we don't want this to be good at replication. This drug will actually fit into a binding pocket in this active site here in the virus RNA-dependent RNA polymerase. Once this binds, it basically just blocks elongation of the RNA chain, the nascent RNA chain, and thereby, essentially interfering with SARS-CoV2 replication. But as I show you here, this is a relatively recent study. Large remdesivir study finds no COVID-19 survival benefit, yet this is standard of care. And if you talk about this on any social media platforms, nobody's going to ban you. However, you're towing the line if you talk about ivermectin or something else, which, one could argue has just as much, if not more, evidence for being successful and efficacious against the virus. So here's a little bit about hydroxychloroquine, which again is an extremely controversial drug with reference to COVID. Here's the virus, and the virus needs to bind, as I showed you, and zinc presumably can interfere with that. But the role of hydroxychloroquine in this particular case is an ionophore, so it allows zinc to enter the cell. And it's really the zinc in this particular case that has the antimicrobial effect. So a combination of ionophore and high doses of zinc might interfere with various aspects of virus replication, such as the RNA-dependent RNA polymerase and also the protease that the virus uses to mature its polyproteins. So here, when the virus is translating polyproteins and then has to cut individual proteins out, there's a protease that it makes in order to do that, and zinc might interfere with that process, and so the virus gets stuck in its polyprotein phase and therefore can't make individual functional proteins. That's what's pictured here. This is the virus protease, PLP2. So here's a quick interview by a clinician in Porto Alegre, Brazil, that talks a bit about using hydroxychloroquine as a ionophore along with high doses of zinc.
What we see is, since we don't have something specific for this phase, even though there is some things, but we're not using for the price, availability, etc., then we need to use a cocktail of things. Just one thing is not enough, exactly because they're not specific. Hydroxychloroquine is very safe and is very old, and it's something that in the medical field, and especially in Brazil with malaria and all that, we are very used to use that. And since used in the beginning, we have lots of articles that show that work. even though it's not 100% work, but work, it makes a difference. And then if we combine this drug with other drugs, then we see a better result. Then that's the reason why we combine hydroxychloroquine with high dosage of zinc. And this is important. Some persons, people end up using zinc, but in a low doses, and then it doesn't work also because we need in a high dose, even though the hydroxychloroquine is a ionophore, it's going to open more channels for the zinc, And then we use ivermectin, like 0.2 milligram.
So what you just heard was the clinician from Porto Alegre talking a bit about their cocktail approach for treating COVID-19. One thing that's been missing in all of this is some studies that can definitely tell us whether, some of the important information that we really need to know, such as infectious dose or when people are transmitting viruses at the highest levels. Again, some of those studies about what the RT-PCR values really mean in terms of live virus, and how long individuals are infected for, so forth, and again, getting back to infectious dose, just knowing how much virus is required in order to cause a symptomatic infection is really important. And so this is finally being done. This is the kind of work that I've been talking about for 2 years now, and there's been a lot of pushback from a lot of individuals because everybody was so scared. But now we're finding that, okay, we can actually do this, even though we knew the data was pretty strongly supportive that we could do this in terms of, the virus affecting extremely old people with multiple comorbidities and so forth. So now we're just taking a bit of another right turn to look at vaccines and the different kinds of vaccines that have been used in this pandemic. This is a representation of the AstraZeneca, Oxford, and Johnson & Johnson vaccine, which are non-replicating viral vector vaccines. And so they use a adenovirus shell to deliver a coronavirus spike gene that you see here. And the inactivated non-infectious particles carrying this bit of DNA is injected into the intramuscularly into the shoulder, into the deltoid, and then the immune response attacks those inert virus particles, and bits of that go into the host cells and are basically expressed. They're ultimately transcribed and translated into proteins that get displayed on the surface of these immune system cells that can then stimulate an immune response. The good thing about this, is these types of vaccines is that they only require refrigerator storage, which makes them a lot more user-friendly, particularly in countries that don't have ultra-low temperature freezers that are required for the mRNA vaccines. In clinical trials at super high efficacy, which is, at preventing serious disease, but I think that this number will probably come down a lot when we look at actual effectiveness in the real world. Efficacy is a clinical trial term where everything is controlled and closely monitored. Effectiveness is a real world measure. How does the drug actually end up working when it's applied to people everywhere and not under controlled circumstances? The other good thing about these is they're pretty cheap, a couple dollars per dose, which again makes them more accessible to poorer countries. The mRNA vaccines, which are pictured here, include the Pfizer-BioNTech and Fauci's NIAID and Moderna teamed up to make this one of the messenger RNA vaccines. So these are just a strand of messenger RNA that encodes the virus spike protein surrounded by a lipid nanoparticle. The vaccine is injected intramuscularly, and the lipid nanoparticle fuses with the membrane to release the genetic material into the host cell. The genetic material is translated into proteins, and then those proteins are stimulating an immune response, essentially, where you see the dendritic cell processing these antigens and displaying them to the rest of the immune response. The neat thing is that this is messenger RNA technology. It's scary in the sense that it's never been really tried before. So this is really a global experiment. But the problem with it is because it's unstable messenger RNA, it needs very ultra low temperature storage. It's also very expensive. And, again, efficacy in the clinical trials is really high, but it remains to be seen what the effectiveness will be in the real world. But I would point out that there's been a lot of pushback from public health about the importance of natural versus vaccine immunity. And I would point out that anybody who dismisses natural immunity and in favor of vaccine immunity is failing immunology 101 for sure. So in vaccines, the vaccine is delivered here, which stimulates a local immune response. And then hopefully that local immune response will pour into the lungs at some point and protect you from a virus that came in and went into your lungs. And so it'll protect you around the back end. And that's why the vaccines seem to be pretty good at helping individuals stay out of the hospital, at least highly susceptible individuals from staying out of the hospital. But it doesn't seem to prevent transmission or infection. But if we look at all this immune response along the upper and lower respiratory tract, you get a sense for why natural immunity should be and is more robust. And so here's various adenoids, the lingual, under your tongue and in the back of your nose and so forth. All this robust immune response that takes place if a virus comes in through either your nose or your mouth, and again stimulates all this immunity. These are M cells that process antigens and stimulate an immune response here and here and here and so forth. And so again, you produce a ton of very specific antibodies here, and that basically creates an essential barrier to infection. And that's why we have flu mist, for example, as a live weakened virus that gets misted into your nose, and as a result will set up a more natural type immune response. But anyway, the point is that the natural immune response, if you happen to get infected, is, in my opinion, far more protective and far more important than vaccine immunity. And it's supported by papers such as this, Science article, SARS CoV2 infection confers greater immunity than shots. This came out, and a lot of people didn't want to pay attention to it because it sort of went counter to the narrative, which was to get everybody vaccinated. Even the CDC at first started to say that, that vaccinations are way better than natural immunity, and now they've been forced to sort of change their tune a little bit. One of the issues that I have with mass vaccinations is this concept of rapid evolutionary escape, meaning that if you use a non-sterilizing immunity type vaccine that's very specific. So again, non-sterilizing immunity means it doesn't, again, prevent you from transmitting. It doesn't prevent you from getting infected. It's not sterilizing. The virus can still replicate in you. It just may not kill you or send you to the hospital if you're vaccinated, if you're a certain type of individual, meaning having multiple comorbidities. So if it's non-sterilizing and very specific, that could lead to a buildup of large standing populations of virus that are more likely to undergo immune escape. And if we look at this graph, this happens to be one figure buried in a Nature Med paper that's linked here. What it shows you is that the potential for myocarditis in under 40-year-olds, excess events on the y-axis versus the particular shot. And so here's the AstraZeneca, and it looks like there's no threat of excess events of myocarditis. Here's the Pfizer-BioNTech messenger RNA vaccine, so there's some. But notice this is the Moderna shot 1 and then Moderna shot 2. This bar is Moderna shot 2, excess events for myocarditis compared to a SARS CoV2 positive individual. And so what they're showing pretty clearly is that you're more likely to get myocarditis if you're under 40 from the second dose of Moderna than you are from the natural infection. And still the authors conclude that everybody should get vaccinated. Let's not, overblow this, but the data are here. And so there is evidence. And so when some people worry, you can't just outright dismiss them because there is evidence for what they're saying. And even entire countries are declining to recommend vaccinating kids, again, because they don't see any clear benefit. If all you can really say is that your vaccine will prevent you from going to the hospital, and we know that kids aren't really going to go to the hospital, or at least a typical, healthy young kid is not going to go to the hospital anyway, what's the benefit? You might actually be exposing them to a little bit more risk. So the last question, how does it end? Herd immunity, natural or vaccine-based immunity, probably a combination of both. The virus will definitely attenuate. That's what viruses do. We have lots of coronaviruses that pass around seasonally, and they sort of have floated under the radar for a long time, even though they kill a few thousand people every year. This is a practical example of that. The 1918 flu, which was an H1N1, a version of it came back in 2009, but it wasn't a major killer. Here we lost, nearly 700,000, maybe a million Americans, and here it was probably 12,000 in a much more dense population. So we attribute this to immunity that developed, by exposure to these viruses over time. Will we come up with a good antiviral? Do we just need modest improvement? Do we need it to be a game-changing killer, a virus killer? That remains to be seen. But again, the drug discovery is happening fairly quickly, and there's a lot of money in it, so a lot of incentive. And lastly, burnout. People just get sick of hiding and being scared and being traumatized, and just decide to go about their lives and accept it as just another risk. Getting into a car, for example, or whatever else. So that's a combination of bullet points on how this ends.